Meeting is being recorded, it says,
Mm-hmm.
and everybody is inside.
Yeah.
Everybody who's allowed inside is inside.
Mm-hmm.
Because when I
Yeah.
click on the gallery view, I just get the three of us, James Lamb and you and me.
Yeah. Because we are sharing our videos right
There we
now.
go. Now we got more people. All right. Now we have at least two. Yes. Okay. Okay. So here we go. All right. So what I got to do is share screen. Hit this guy. There we go. All right. Now, today we're launching into unknown territory. This lecture and the one to come on next Tuesday are both brand new lectures for me. I've never given them before. Um, they're put together out of slides from a variety of other lectures given for other purposes to other people. Whereas the ones we've been doing so far are ones that I have given in general for this course previously. So these will be on Zoom, on Canvas, just like the others. But there's no images on the University of Pennsylvania website, something you could go to read to get more background information about them. So the only thing we have here will be the actual slides and the comments that I make and the papers that go along with them. So this is a little different for these two. And because they're, for me, new, I'm approximating how long it's going to take me for each one, which determines how fast I can go for each one, okay? Now the topic for the first one, well, they're both, the reason why they're put together like this is that as I started building these two lectures, I realized that they're so totally intertwined that I have trouble thinking of them as two separate lectures. So what I did was make a very long lecture and cut it in half. And so the first half is what we get today, and the second half is what we get on Tuesday. Now, it's all about names. And in the science world, names are taxonomy. And in the whole organism world, taxonomy usually is dealing with whole things, whole organisms, whether they're elephants or whether they're an ant, they're something. Now, in the chemistry world, which is also science, of course, and the molecular biology world, there's also a lot of taxonomy of the things they work with. But the rules for those taxonomies are quite different than the taxonomy we will be referring to in and out during today and next Tuesday. Anyway, the general topic here is what's in a name. In other words, what does the name represent and where does it figure in what we think about and what we do? Now, taxonomy for classical taxonomy for organisms is basically just the practice of putting names on things. But it was focused on, still is focused on, stability, extreme stability, meaning that once people put names on things, they didn't want the name to change. Now that's actually quite a hopeful thought, which is not met in many, many cases, but they chose Latin as a dead language to be the root for the, or Greek, to be the root for these scientific names because they thought a dead language, of course, would be dead and therefore it wouldn't mutate and change like our modern languages do. So that was their goal, was stability, and that then generated a whole set of attitudes and rules around a name. And we're going to touch down here and there on those rules as we go through. This will not be a study of a rule book, okay? Then the next on Tuesday, we'll get more into the use of those names, but there'll be a lot of the use of those names today as well. Now, next one here. Why am I not going forward? For some reason, my PowerPoint is not advanced.
You might need to click on the PowerPoint.
might need to click on the PowerPoint. Click on the PowerPoint.
What do you mean, click on the PowerPoint?
That you advance on the PowerPoint.
No, that's what is not advancing.
No, but click on the screen.
On the screen. Oh, okay. Yes, it did. Okay, got it. Thank you. And we can do this way. Yeah, okay. So now it's working. Thank you. Now, taxonomy goes back to a very famous person called Linnaeus, a Swede back in the 1700s, who first came up with a plan in the sort of the scientific guild, the scientific level of people, what was designed to give stable names. And so the classical way of dealing with taxonomy is to start way back then and walk your way forward. That doesn't work well for me. It spends a lot of time on stuff that isn't relevant anymore, unless you're a historian. I'm more inclined to start now and work my way backwards as it may be useful here and there. And the reason why for that is that I'm focused on today's situation with taxonomy and the organisms involved and their future, not on what was going on in 1950 or 1920 or 1850. And my goal is not to recreate the past. Unfortunately, because of the stability drive in taxonomy, a lot of people would like to recreate the past. But that past does not fit with the very, very fluid and changing world that we're in today. Now, my target, the thing I'm focused on, that then gets focused on in this lecture as well, is a thing called BIOL140. And you're going to learn a lot more about that one word as we go along. And its analogy comes from the word literacy, which of course you all know, reading and writing. And bioliteracy would be reading and writing about biology or about the things that biology is, okay? So I speak of reading the forest, as in knowing what is going on in it when you walk into it. Well, I work in Latin America. I don't work up here. And so it's all in Spanish. So alfabetismo is literacy in Spanish, means reading and writing. As you can see, just a second here. This is reading, leyendo, y escribiendo. Escribiendo is writing, all right? So that's just straightforward, same thing as you see up here. Bioalphabetismo, on word, putting bio in front of alphabetismo, is reading and writing biology. So it's really direct translation of what you see up here. So bioalpha comes from this first part of that word. So we needed a word for this process that we're going to be talking about all this time. And bioalpha is the word that has sort of emerged very recently. Now, to put it in one sentence, it's to render the 5 million people of Costa Rica to being bioliterate. In other words, the 5 million people in Costa Rica, that's the population of Costa Rica. It's smaller than Philadelphia, in other words. And my goal as a practical outcome, and we're not talking theory here, we're talking actually doing it, is to render those 5 million people not fully bioliterate, of course, but to get them started on their own that way, the same way you started with literacy. Somebody taught you to read in first grade or before first grade, and you discovered books, you discovered magazines, you discovered libraries, and you did all kinds of stuff yourself with that discovery. Well, the idea here, of course, is that for bioalphabetismo, if we get the system going such that people find it to be advantageous to them, then they carry it forward as well. So my goal about bioalpha, why do we want to have 5 million people bioliterate? Well, in one sentence, the answer for that is that a bioliterate population of people are more inclined to see something in wild organisms and the ecosystems they live in as being useful to their society, to some piece of their society. So that it gets to matter to a doctor, to an engineer, to a school teacher, to a gas station owner. It doesn't matter who they are, but some piece of biology becomes important to them. And so therefore they're willing to say yes instead of no when conservation aspects come around. So that then settles down into sort of where it is And then how does it apply in conservation? And then where does it sit at the final end of the day after we've done a lot of thinking about it? And these two lectures cut across through the middle of this. We're going to get to conservation and then we're going to stop as well and go on Tuesday. So we start with the real estate. This is Costa Rica right here, little tiny country. Here it is transposed on the United States to give you some sense of size. The size of West Virginia, if you want an idea of how big it is. And it's very, very small. So the real estate that we're working with here with the 5 million people is almost trivial in terms of area. It's just a tiny fraction of the state of Minnesota that I grew up in, for example. But if you count the biodiversity, meaning the things that live in Costa Rica versus the things that live in North America, 4% of the world lives in Costa Rica. 4% of the world lives in North America. So in other words, the biological resource with which to play and to worry about names on and to worry about taxonomy in this little tiny country of 5 million people is the same as it is in this monster up here with its 350 million people or whatever we are at the present time. So we're started there. So here's what the country looks like a little closer up. The green areas are the wild areas. It's roughly 25% of the country. And the yellow areas here are what you and I would call agriculture or agroscape, meaning pastures, plantations of one sort, urban circumstances, roads, all those kinds of things. And so the country is divided basically into two categories of land use. This one here and this one here. And my take on this is that these yellow areas here have got about 100,000 years of practice behind them as to how to use them by people. I didn't say use them well, but they use them in some way or another. And they think they're valuable to them in one way or another, and they don't want to get rid of them in one way or another, all right? That's the yellow part. The green parts are to society, not even kindergarten. In other words, society has walled them off and not really thought hardly at all about what value they are to this entire country, to the 5 million people that are there. This green area walled off came about not through Costa Rican themselves, which would really be indigenous people, deciding that they wanted to save these areas and turn all the rest into agriculture. This is the result of European colonization that took place in the 1500s and has continued up to some time. Now, what's in those wild areas? Well, you've already heard some of these words before, but there are three kinds of ecosystems really in the terrestrial part. There's one more in the marine, of course, but in the terrestrial part, there's what we call dry forest. And this is meant to show you really what dry forest looks like. This is in the dry season over here. Trees are not dead, they're simply dormant. This is like the winter up here. This is the same photograph in the rainy season. And that tree that you see right there is the same tree as that tree that you see right there. So in other words, this is dry forest, and it goes about 6 months looking like this and 6 months looking like this over here. So that's one kind of ecosystem. The second one is what we call cloud forest. We'll see a little more of this later on, but here it is up here in the clouds. In other words, as the dry forest down here sweeps up in elevation and gets up into here, it's covered over with clouds nearly all the time, or it was in the old days, covered over with clouds. And that creates another ecosystem, a whole different kind of ecosystem up here, which is perpetually green, perpetually wet, and has very different dynamics than does the dry forest down here in the lowlands. In this photograph, the yellow is all old pasture. So that would all be agroscape. And then the forest is the dark stuff that you see there. Now you go over the top of that mountain, come down the other side, and you get what's called rainforest. This is basically evergreen all the time, and it rains all the time. So it's wet, 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 wet. Now with climate change, I'm going to go back here just a little bit. With climate change, you notice I said cloud forest was. The point is a lot of this up here now is disappearing because with climate change, all of this down here is getting hotter. And the hot air rising up through here melts these clouds. So this becomes cloudless or less cloudy, much longer, many more days of the year. And that results in, of course, drying out this very perpetually wet forest up here and perpetually dries out the amount of water that flows out of them down into the lowlands. And then, as you say, we go over those mountains. This happens to be a cloudless day, so you can see the actual tops of the mountains up here. This is an active volcano. That's why you don't see any forest on the top. And then down into the rainforest, which extends all the way out to the Gulf of Mexico, to the Caribbean coast, in other words. So this is evergreen all the time. And as I say, rains all the time. So those are the three major ecosystems. Now notice I used a word in each of these. I called this rainforest. That's taxonomy. That's assigning a name to it so that when somebody in Brazil or Venezuela or Africa or Southeast Asia is talking about their forest, they will use the same words. They will say rainforest or cloud forest or dry forest. Now, in India, people have a tendency to call dry forest monsoon forest because they call the coming of the rains a monsoon. So the outcome is that the same phenomenon is going on, but it gets its taxonomy changed, which creates all kinds of confusion on the street and in legislation. Because people talk about dry forest in the New World tropics, they talk about monsoon forest in the Old World tropics. And newspapers and reports about hurricanes and all these kinds of things get very confused around these two words, which is part of the problem of taxonomy and why people like to keep one name for one thing as long as they can. Now, this is my laboratory. We were just looking at dry forest, which is all of this here. Cloud forest is up on the tops of these volcanoes. And the rainforest is over here on the Caribbean side, going out to the Caribbean over there. And this is the Pacific Ocean down here. This is the size inside the white area of Philadelphia. So this is my lab, all right? Just like, you know, the molecular biologist got a lab on the third floor lady, and there are machines, there are desks, there are graduate students, there are undergraduates, there are all kinds of things doing things. Well, this is my lab. And as I say, it's the size of Philadelphia, and it's the ecosystems, the three ecosystems inside of that. And each of these red circles is effectively a machine or a lab or a desk in my lab. And my desk is this one sitting down here in the middle of dry forest. So this is where Winnie and I live for 6 months of the year. This should give you a physical idea of what it is that we're actually talking about for doing these two lectures. And these are my students. These are basically, there's one PhD in that photograph, two people who got master's degrees. The other half of them are people who made it through high school, and the other half are people who maybe made it through 6th grade. And they are, in contrast to you all, career parataxonomists. You, if you were to go and volunteer to be part of this during some time in the year, you would be volunteers. You would be students passing through. These people are people who were in the working class agricultural landscape and have moved out of that to do what you do full-time as a paid career. And they're all residents, they're all Costa Ricans. And what they're really doing is biodeveloping. Notice I stuck the word here, bio, on the front of the word development of a conserved wildland. So what does biodevelopment mean here? So now we're back to taxonomy again. What it's referring to is doing anything with that forest that you would call development, like you would say agricultural development or internet development or highway development or health development, but you're doing it with that forest without destroying it. So you're doing it in such a way that it doesn't damage the system, okay? And taxonomy is a huge piece of that, because all this greenery that you see up here is not just greenery. That's the organisms. Those are the things that are actually doing things and that you then, the parataxonomists, are working with. These are the lab rats and the mice. They're everything. 650,000 species of them live there. That's about the same number of species as in 2/3 of North America. And they're sort of everything, from whales in the ocean, to a cockroach on my thumb, to the face of a caterpillar over here. This is a tapir, a deer-sized, overweight deer-sized mammal. Little tiny things like mosquitoes. And the idea is that these lab rats and mice continue to be there forever, into perpetuity. So the goal of this system of biodevelopment is that it doesn't exhaust it. It cannot afford to go bankrupt. It cannot afford to be taken down to the parking lot for a mall. It has to stay in itself, in its own dynamic self. And this is like I say, these are the organisms that are involved in all of that area the size of Philadelphia. So me, as a hardcore academic, ivory tower, famous in quotes, academic researcher, landed in that back in 1978. Well, actually, I got there in the 60s, but they really focused on it in 1978. And my research goal, just like somebody's lab in Leyden has a research goal, my research goal was to find all the species of caterpillars that live in that place. Not only find them, but find them in the sense of getting a name on them, find them in the sense of knowing what they eat, finding a sense of who eats them, who is a parasite of them, how long do they take to develop, what good are they useful for in any way or another. In other words, everything about this animal. And that was a very academic goal. My grant proposals to the National Science Foundation in the beginning stated in the beginning, there are no practical applications of this research. And in the 70s and the 80s, that was viewed as a very acceptable way of going at it. Okay. Now, we knew there were a very large number of species there early on because we hung lights out in the forest. This is powered by a car battery. And then that attracted moths. So you look at the moths and you count how many different things there are on that sheet. And you realize that behind every one of those was a caterpillar. In other words, there was one of these guys behind every species on this sheet. Now, the striking thing to me was, you say, well, so there's caterpillars, and we know about caterpillars in Wisconsin and Illinois and Virginia and Florida and so on. That's certainly true. But in the tropics in 1978, and still today in almost all of the tropics, you step out of your airplane into a tropical forest and you find a caterpillar, and you turn around to anybody in the science community, the museum community, and you say, oh, what's the name of this? So I can look it up in Google and find out about what we know about it. And nobody can tell you the name. If you have a moth or a butterfly, one of these things in your hand, somebody is actually likely to be able to do that, certainly if it's got any size to it. Right? But the caterpillars, completely unknown. This was just a great void in information. Basically, why? It's because when you collect something like this, it decomposes as soon as it dies. You can't save it. Whereas the museums of the world are full of these things, saved as specimens on pins, okay, and then worked with for their taxonomy. Now, let me give you an idea of how you get a feeling for not only the light trap that you just saw. This is called a light trap, incidentally. You put the light up there and the moths come and sit on the sheet, and you walk up to the light, and there you can look at them and think about whatever they are. There's another thing called a malaise trap. This is a malaise trap. And if there was a person standing here, their head would be right about there. That's how big it is. And it's like a tent with the door open. Like, think of this as the open door, and the tent is inside that. And there's one from the other side as well. So insects come flying along, go into here, hit this partition in the middle, and try to escape by going up, by flying or walking up. So they go up here and they get narrowed into this, and there's a hole in the top here, and the hole enters into a plastic bottle, which you can see right there, full of alcohol. So they drown drunk. In other words, they're killed for a few seconds by dropping into this alcohol. And what you do is you come by 1 week and collect the bottle. So now you have a sample of what was flying back and forth through here. So this, incidentally, this is cloud forest as opposed to the dry forest you saw down below. And you can in fact see the clouds up here. See this, all this misty white stuff here. Well, these trees live in this kind of world all the time, and there's water dripping down the tree trunks and so on. Well, if you do this in cloud forest for 1 year, you put that trap out there for 1 year, right there, and you put another one right here in the middle of the dry forest, and another one right over here, should be right there, in the rainforest, you do that for a whole year, you get this kind of result. The dry forest one caught this many species in a year. The rainforest one caught this many species in a year. The cloud forest one caught this many species here. And when you add them all up, it's 17,000 species in an area about the size of the campus of the University of Pennsylvania. Maybe a little bit, maybe twice the size of the campus. Now, that's an unbelievable number. Nobody wants to believe that, all right? But it's real, this is just data. But striking to me, not only is the enormous number, but in common between these 3 different traps, there's only 31 species. In other words, only 31 species occur in all 3 ecosystems. That tells you that each of these samples, this sample and that sample and that sample, are drawn from a huge pool of different species. So when you get all done and you do inventory like this for year after year after year, and you add up how many, you're gonna, this number down here is going to be 100,000 or 200,000. It's going to be a huge number. Now, how do you deal with all those different pieces? Put it together in your head and think of how you're going to deal with 200,000 different kinds of food in a supermarket. You have to have names on everything. Now, for centuries, humans have dealt with things by just putting names on things. You go to the supermarket today, there might not be 200,000, but there might be 20,000 in a big supermarket or more, and there's labels on them, so you know what it is inside. Well, what's the label you're going to put on all these different insects to figure out what they're doing? Okay, and once you've figured it out, keep track of the information and then be able to transmit that information to other people, both distant-wise geographically and through time. So 100 years from now, they don't have to go out and catch the same thing over again and figure out all the same things that you figured out. Okay? That's where we're headed, is to that challenge. So in effect, this trap, which catches these insects in a bottle, is like a rain gauge sitting out here in the agricultural landscape, which is catching raindrops. And it pools the raindrops. It gives you information about how much rain was falling. This pools the insects and gives you information about how many insects there were. And furthermore, you can do chemical analyses of the rain that's falling and tell you whether there's air pollution going on, whether it's summertime or wintertime. There's a whole series of things you get out of this little automated rain station here. Well, the same thing here. This requires nothing but putting up this little funny little trap and putting a bottle of alcohol and catching a very, very large number of insects that come along with it. So what the analyses that people have been doing with meteorological data for like forever over here on this side, by psychological analogy, this is being done with these things over here. So inside this bottle might be 200 species of insects, most of which do not have names. Now, how are you going to deal with that? Most of which do not have names. And to you, they're just ugly bugs. So the question is, we're going to get to that, but that's the challenge that we've been dealing with for hundreds of years. Now, the way we deal with that now, after 2003, not before, after 2003, here's all the bugs that come out of one of those little bottles on the trap, right? Beetles, flies, grasshoppers, all kinds of things. You pick out one of them under the microscope and you take one leg off. And from the one leg off, you put it in a little plastic well right there in this plastic plate, which has 95 of these little holes in it. So what you're doing is you take the leg off of this insect, you put it right in that hole. You can see there's the leg going in the hole right there. That little plate then goes in a robot, and then the robot extracts the DNA from that piece of a leg, from a very, very small piece of leg. So what you're doing is you're taking all those insects from all those traps and any other way you catch them and shoving them through this, and it's not dealing with a name, it's dealing with the code of that little well in the plastic plate and the DNA that goes with that little well. So you have the bug now, and then you get DNA from a mitochondrial piece of the genome. All right? So here's, just to remind you, you have a genome in a cell, which is basically the big chunk of genes that you run around with. And then you have a small mitochondrial piece of the genome, which deals with just a few essential pieces of your metabolism. This is actually a microorganism that parasitized our lineage a long, long time ago and helps us and deals with oxygen metabolism. And right here is a gene called CO1. And what it's doing is this is about 1,000 base pairs long. And what it's going to do is grab those 1,000 base pairs, and out of this end, it's going to take off 650 of them. And so we call that, by analogy, a barcode. So by analogy, what I mean is, a barcode, is that this little 650 base pair piece is the same as the barcode on a can of tuna fish in a supermarket. In other words, you can look it up and see what the database tells you about something that has that little code. And in this case, somebody has taken the insect beforehand, or maybe you do it yourself the first time, and you get that little piece right there. And that then becomes the barcode for that species of insect. Because everybody listening to me has got the same barcode. Every one of you. And you've got the same barcode as somebody in Australia, and somebody in the North Arctic Circle, and somebody in the Sahara Desert, and somebody anywhere. And yours is different from every gray squirrel on the East Coast of the United States. But every gray squirrel on the eastern coast of the United States has got the same barcode. So here's what it looks like. So the way to imagine this is a DNA barcode is a word made up of, sorry that this is in Spanish, I give these lectures in Spanish a lot, and so sometimes these creep in. A word with 600 letters. So this is 600, it can be 650, 640, and an alphabet of 4 letters. So if you've got a word that looks like this, you can go and you can change that one right there, and that one right there, and that one right there. And that makes it different from every other barcode on the planet. So this is the actual barcode from this actual fly. The way we got it was you take one leg off. Okay, all insects have 6 legs. All of our insects have 5 because they've all lost one leg for this process. Now there will come a time when we'll be able to get this barcode without having to take a leg. That's a hair will be enough, or a little spit, or just any piece of tissue from this fly. But at the present time, at a pretty crude level, we're still taking a whole leg, and there's a lot of DNA in a leg, and that's enough to get a good solid barcode that looks just like this, and that's what's used to tell these things apart. So this fly at the moment in time has no scientific name. But what it does have is this barcode, and the way you get to that barcode is you use this voucher code right here. So that code right there takes you to a database which has this in it, has that in it, and has the whole history of catching this fly, where it was, what it was doing, how you caught it, and so on and so forth. Okay. And from those traps, we get enormous numbers of insects. So for example, here's a trap that caught 80,000 individual insects in 1 year, and now we barcode all of those 80,000. So now we can say that there were 10 individuals of this species, or 23 individuals of that species, and 1 individual of this species, and 223 individuals of that species. Okay. This is all done by robot. So now, what do you do with these traps? The esoteric scientist comes along and says, oh, I want to know what's the difference between dry forest and rainforest. So you do that and you find all that, but then so what? Well, you see that white spot right there? That's a Malaise trap. And it's been sitting right there all the time that this entire site for a geothermal drilling operation was carved out of this rainforest. So this was a gorgeous, beautiful piece of untouched forest. The country itself needs to tap into the geothermal resource, which is underground, very large quantity of hot water. Bring that up and use it to run turbines. So the question becomes, what kind of perturbation happens to this forest? If you cut this big 2-hectare hole, maybe about acre and a half, 2 acres, something like that, hole out of the forest, and do this geothermal drilling here in the center, and set the thing up and then walk away from it. Well, this trap has been sitting right there since the first chainsaw was applied to this forest. So we can monitor how the insect community changes over time as we go along. We're going to talk about that more in the second lecture. But the point is, I want to say that this is not just esoteria. This is stuff that you can actually make the forest, the insects in the forest, have a value to people, because people want to measure this to understand the impact. But if they can't read the forest, if they're not bioliterate, they can't understand the impact. Now, traditionally, things that you find in the wild end up in a museum. This is the National Museum in Costa Rica. Excuse me, it's not the National Museum, it's the National Theater in Costa Rica. And these are butterflies, as you would see in a museum. By my analogy, these are, in effect, part of the National Theater of Costa Rica, because they're what people in their urban world look at, which is what people see in the movies, in the theater, in an art institute. Okay? So these are basically proxies for what's out there in the field. So the real living things are here. But you can't take the whole herd of deer and put them up in here to get the urban people to understand it. You can put one there, but you can't put 5,000. So what you end up doing is viewing the national park as a kind of national theater also. But the things that are in it are alive. So when you walk out in the forest and you see this deer, are you just saying, well, oh, that's a deer? Or are you saying, oh, what I know now is that's a male deer. And I know that he's just growing his antlers, because you see this fuzz on the outside here, it's called velvet. That's an indication that these are the antlers which are still growing for the year. So in another 2 months to 3 months, he will have antlers big enough to polish, make hard, and he can fight with other male deer over female deer. We can go on and on. We can talk about these gorgeous ears, which are, of course, aesthetically beautiful, but also part of him keeping very much alive, because he's extremely good at hearing what things are going on around him. And the tongue and the nose and the eyes and all these things, you can begin to, but if you understood them beforehand, then when you see them out here, you can read it. You know what is coming out of that animal, what that animal represents. Now, traditionally, for hundreds of years, we have collected these things here and taken them inside our museums and stacked them in cabinets. pardon me again, it says over here, we are drowning our museums in witnesses. These are the organisms that were caught by people in traps in their hands and with butterfly nets and with lights in the forest and all that kind of stuff. And they're put into drawers like this. There's huge biological knowledge in this thing locked up in these drawers. And the only justification for them is they're pretty. And we don't want to lose them. And there's a whole set of people who make a living and specialize, and they're very smart people, and they're very good people, specialize at simply studying these things, like people moving things around in a rare book library. After 2003, the world changed. And the way it changed was a very funny sort of way. This is a very common butterfly in Latin America. It doesn't occur, well, it occurs in Texas, but it doesn't get up to this part of the world. Everybody's seen them on flowers in their garden and so on. This is a very standard thing. And these are its caterpillars over here. His name is Astraptes fulgurator. And in 2003, this is John Burns at the Smithsonian Institution, and he's a sort of world specialist at understanding how to tell these things apart, put their names on them, do taxonomy with them by what they look like. Okay? So we looked at these drawers of this butterfly, many individuals reared from caterpillars. And he said, Dan, you know, I think there's more than one kind here. In fact, I'm pretty certain there's more than one kind here, even though they all look alike. But the differences are so subtle that I can't consistently say, this one is this, and that one is that, and that one's that one. I can't put a name on this, that, and the other one. Now, all of them were going on one name, Astraptes fulgurator. This is taxonomy now, hardcore taxonomy. That is the name for the thing in this family of butterflies. Okay. And you would look at this and say, you know, I'm pretty certain there's more than one thing. I thought there was more than one thing there because these things, these caterpillars over here, feed on many different kinds of plants. And butterfly caterpillars in general do not feed on many different kinds of plants. Each butterfly species in general has its own set of food plants. Whereas this particular butterfly had this horrendously long list of 53 species of food plants that its caterpillars were feeding on. Well, by accident, Winnie and I were invited to a molecular biology genetics kind of conference in Cold Spring Harbor in the eastern United States. And we were bystanders, basically, just meant to listen. And we witnessed a huge argument between these two professors. This is Professor Paul Hebert from Canada, the University of Guelph. And this is Dave Hillis from Texas. And Dave Hillis and others at this meeting were all highly focused on the human genome, getting the whole genome, the whole package of genes. Because for obvious reasons, human beings care about that whole thing for many obvious reasons. So that was their focus, as to how do we get the whole genome and know what the different genes do, right? Paul came along and said, well, wait a minute. All I want is a little piece of that mitochondrial genome, 650 base pairs out of the billions, just 650, that I can use to tell them apart, to identify them. And Hillis was a member of the group of people who said, oh no, first off, that's silly, that's kid stuff. We can tell them apart all kinds of ways. And furthermore, we don't want to waste our time on that kind of stuff. We're after the big thing, the whole genome. And Paul was saying, yeah, but we can use these other things for identification. But the people who cared about whole genomes didn't care about identification. They didn't care whether you could tell one cockroach from another cockroach, whether you could tell one beetle from another species of beetle, whether you could tell one species of bird from another species of bird. Those things didn't matter to those people. And I can understand why. But to Paul, getting that little piece of the genome was everything. And it was a very disruptive meeting. And at the end of the day, no conclusions were arrived at all. Well, I'm listening to Paul talk about this methodology. And I remembered back to high school reading science fiction books way before Star Trek. And way before Star Trek, there were science fiction books who talked about people landing on other planets and pulling something out of their pocket and pointing it at other things, and the other things would, and the gadget would say whether they were edible or not edible, or poisonous or not poisonous, or dangerous or not dangerous, and all those sorts of things. The sort of the thing that Star Trek came up with much later. I always thought that was one of these science fiction dreams, and didn't have any base in reality. I still don't think of it landing on another planet as having much base in reality, but I'll leave that to one side. The point being that after listening to Paul talk about this little thing, I stood up and pulled my comb out of my back pocket and held my comb up like this. And I said, Paul, I want to see what you're talking about. It's something that costs what this is. A little cheap thing that everybody in the world can have. Because what Paul was thinking about is you get the sample, you get the leg, you get the piece, the blood off the airplane, whatever it happens to be, and you send it to his lab. And he does the analysis there, and then he gives you back the results by email or whatever. What I saw immediately was, what mattered to me was, how do we get this in the hands of everybody in the planet so they can become bioliterate by being able to read anything, anytime? So he looked at me and he said, well, you've raised the bar. And I said, well, yeah, but that's the only bar that counts. So he went home and back to Canada. And we invited him down to the Smithsonian Institution to come and give a talk to taxonomists, real hardcore taxonomists, these people who make their living off of being taxonomists, off of moving and shuffling and thinking about those insects in those drawers and giving them names and writing up papers about those names and writing papers about the insects in their drawers and all that kind of stuff. So Paul gets up on the stage and he gives the same talk about, hey, wow, you can identify things this way, rather than the way we were looking at here, rather than looking at them. Now, if you spent 50 years of your life doing this right here, looking at this, and you're the only person in the world who can do that, what is going to be your reaction to somebody in a big major institution that stands up on the stage and says, you know what? With a barcode, I can tell anything apart, which means every school child can also tell everything apart. Suddenly, your prominence, your position in society has vaporized. If you're the only person in the world who can identify every scorpion in Africa, and suddenly somebody comes along with a gadget that every school child in Africa can have in their pocket, that they can identify any scorpion who's just stung them, suddenly you are not happy. If you're a young PhD and you just learned this and you haven't spent 55 years of your life, look at it and say, wow, imagine the things I can do with that. So we had two very different reactions that came from the taxonomic community, the people who spend their lives doing taxonomy. So I said to Paul, we have this drawer full of insects, which we think might be more than one species. Can we give you a leg, take home? So this is 473 butterfly legs, each one in its own Eppendorf tube, smuggled across the border into Canada for DNA analysis. And so here's a box of those butterflies. And as you can see, they look very, very similar. Now, if you stare at them for a long time, you can begin to notice little differences. In some cases, this white area here, for example, extends way down like this, where here in this case, it's very short, right across there. But it's a little longer here, it's a little longer here. And you start looking around, you find intermittent, here's one where it goes way down. So the consistency in telling these things apart begins to vaporize. Well, in fact, when you DNA barcode the legs from all of these butterflies, there are 9 species of butterflies in that box. And what it boils down to is, pardon me, oh yes, part of this is in Spanish. This is the Costa Rican newspaper here. This is the English one. Basically, the barcoding reveals what the taxonomists call cryptic species, meaning that they're very hard to tell apart. And now we can tell them apart by the barcode. And if we go back to here, we suddenly discover that each one of them feeding on a different species of plant has got a different color pattern in caterpillar. Now I feel really stupid because I collected all the photographs for all these things. But you get one now, and one next year, and one 3 months later, and one and I had never put them all together. To notice that all the ones from Onchocarpus had this color pattern. All the ones from Hempea had this color pattern. All the ones from Bitnaria had this color pattern. So what happened, turned out, is that the adults are basically identical. And to a bird chasing the adult, they look exactly the same. But to the bird foraging for caterpillars, he sees different color patterns. Now, in fact, you now know enough as guess what all these color patterns are. They're all coral snake mimics. So to a bird, that's a coral snake. Red, black, and yellow. Costa Rica became very proud of this. This is their New York Times of Costa Rica, La Nación. And here's the butterfly. They took that photograph in their own backyard. The newspaper people did. This is one sitting on my hand, but this one is here because, like I say, this is it's like the monarch to them. This is one of the butterflies that they've all seen in their lives. So this caused us to start looking hard at the web. Now, when you go on your ecotourist trip to Central or South America, and you go to your fancy lodge and you're in an eco lodge somewhere out there in the rainforest, they will commonly put out on a banister somewhere some rotten bananas to attract butterflies who come from the forest to feed on your bananas. Well, here's one. There it is right there. This is taken directly off of Flickr. And the classical entomologist who's enlisted by somebody to either by the commercials or by the person, the amateur who took the photograph, to give them a name. So they give them a name, and they call it Propona laertes right there. So this, the proud person, this guy here, right here in 2010, put this butterfly photograph on Flickr, and people will take that photograph and they will, as it says, this photo also appears in Costa Rica Butterflies, Tropical Entomology, Butterfly Color, Natural Costa Rica, Butterfly Blues, and probably another 35 other places, all with this name. Well, oh, incidentally, here we are. This is where it was taken. Here is Costa Rica right there. But we don't catch them with a butterfly net or with bananas. What we do is rear them from caterpillars, because that's what we try to figure out, is what the caterpillars are. So we get all these adults without knowing what I'm telling you. But we have a lot of adults, which we save as witnesses, as testigos, as vouchers, as we say, as vouchers for the DNA. They're all barcoded. And suddenly you discover that each line here is one barcode. So that's one caterpillar right there. Here's another one, another one, another one, another one. All these caterpillars have got the same barcode. All these caterpillars have the same barcode, but different from this. So here's one barcode here, another barcode here. Both of these, anybody would call Propona laertes, just as you just saw back here, Propona laertes, off the web. There's the butterfly over here. This is a reared one over here. This is what they look like. I mean, you tell me how to tell them apart. These occur in the same square meter. We're not dealing with a zebra in Africa and a horse in South America. These are right together all the time. However, all of these caterpillars here, we're feeding on bean plants, Fabaceae, the family. Now notice I'm using a name now. This is taxonomy. I'm using a name that everybody in the world would recognize, any scientist or agricultural person would recognize in the world is going with beans, peas, lima beans, garbanzos, all those lentils, all those things are in the Fabaceae. So that's a name that was applied. Everybody wants to keep it that way. So if you come along and you name a coconut Fabaceae, the whole world will fall on you and say, whoa, no, you can't do that. Okay. It's an unspoken regulation. All of these are feeding on a different family, Chrysobalanaceae. It's a tropical family that you wouldn't know about. But the point being is these two plants, Fabaceae and Chrysobalanaceae, can grow in the same square meter. So that mom knows them apart. She comes in and she puts her egg on one or the other, depending on which species she is. Okay. Now, the striking thing about taxonomy is, I'm sitting here looking at all this, and I look at the names, and Laertes is the name that everybody has, and all the museums in the world have for this butterfly. And I discover, like with Google, that back in the late 1800s, this butterfly had two different names. Two different names. From entomologists, taxonomists back in those days. So what they did was they looked at these butterflies then, and they came to the conclusion that there were two species. Now, how did they come to the conclusion there were two species? You see, right here is a little yellowish patch, right there, and right there. That's a little plume of hairs, right here. Now, when you have a freshly reared specimen, a new specimen, it's just come out of the pupa, one of these two species, I can't remember which one, has a slightly more yellow patch of hair, and the other one has a slightly more orange patch of hair, just ever so slightly. But now that I know there's a difference, I can tell them apart by those two colors. Well, in the 1800s, the specimens were freshly caught from South America and Central America, sent to England, sent to Europe, sent to Germany, to France. So they were working, the taxonomists in those days were working with very fresh new specimens. They mounted them, they put them in museums. Over the years, over the decades in the museums, the color of these little plumes hairs fades. So now you go and look at these specimens collected 100 years ago in the Smithsonian Institution, and all of them look exactly the same, with the same color in these hairs today. So today's taxonomists came along and looked at the two different names in the two different drawers and said, oh no, these are the same thing. So they synonymized the two names. So now in the literature, these two different species are operating under one name. So we come along and discover that one of them is feeding on Chrysobalanaceae and the other on Fabaceae. So we know that these are two different species here. So now what has to happen is somebody has to do the taxonomic legal work of resurrecting the old name from back in the 1800s that was, as we say, sunk under the new name. So now they go back to having two names again. So now if you look at the most modern databases, this species of butterfly, which looks like one thing, in fact, is there under two things. But now the museums and the taxonomists complain bitterly because they can't tell the specimens apart. They have to barcode them to know who's who, unless it's the fresh caught one. But if it's something's been in a museum for a long time, so that puts them up against a rock, between a rock and a hard place. They have trouble telling the things apart. So they find it very inconvenient to use DNA to tell things apart until the day when they have their own comb in their back pocket that they can use, like a school child can use, to get the DNA from a sample. that time, then suddenly things get a lot easier. But right now what we have is a science community which divided. There's a set who find this to be very useful, very refreshing, very helpful, and another set who's very antagonistic to it because it makes it much, much harder for them to deal with species. Just to give you an idea, these are the genitalia taken out of the rear end of a pair of butterflies that are identical to your eye, but you can tell apart by barcoding. Now, these are very, very carefully done, very good drawings. So any difference between this pair here and this pair over here is real. So if you look at them, you see that there's a bump right here. That bump is not over here. Just a slight depression right here. And the proportions across from here to there are a little different from across from there to there. Now, taxonomists in the old days, looking at this, would say, oh yeah, these are the same species, because that's just slight individual variation. It's just a slight difference between the way that, you see how these are curved out here like this, whereas they're more straight and not so neatly, not so pointed and curved away. So they would say, oh yeah, that's just variation. What happens is you come along and you barcode a whole lot of specimens. You discover that there's one barcode goes with a whole bunch of them that look like this, and a barcode goes along, the other barcode goes with ones that look like this. And you realize, oops, we got two species here. Okay. These are the butterflies that I was just showing you. And a very interesting thing about those of you who work with genes and genomes is this. These are the individual, that's the individual butterfly right there. Each one of these is different. And this, see it says right here, 658, that's the number of base pairs in its barcode right there. Now here's one down here, it's 379. This is very, very short barcode. And there's another 300 one, another 300, whoops, sorry, there's some more of them here. Yeah, there's more 300s there. There's a 300 here, here's a 300 here. What we know is that's where it lost a piece off the end of the barcode. So that's how it got shorter. When you compare those barcodes, they're all the same. There's no difference at all. When you throw out the short ones and you work only with long ones, 500, 500, 500, 600, 500, 500, 600, all of one species groups together and all of the other species groups together. So what tells you is that the diagnostic piece of the barcode was what got chopped off when you got short. So these means that when you're doing your extraction and you've got to count up those base pairs, it takes care so that you can't just use a little short piece of sequence. You've got to use a long piece, long enough to have gotten the difference between the two. Which, look at the two butterflies. They're very, very, very similar. Their caterpillars have slightly different faces. So here's one, this is one species here, this is the other species there. But you see it's reddish here and more yellowish here. And the sort of the gestalt of the face, the way the black V goes all the way down to the bottom here and only goes part way down to the bottom on this side. They eat the same food plants, but this one is in rainforest, this one's in dry forest. And where the rainforest and the dry forest come together like that, you can have both of them on the same plant, right side by side with each other, but they don't interbreed. Now, speaking a little bit more about the magic of these barcodes and how this relates to taxonomy. This is one species, that's the underside, and this is the top side. This is one species of butterfly up here and another species of butterfly down here. Okay, so underside, top side. And you notice that up here, it says Adelpha malanthe all the way down here. See this line that comes down here and goes right down there and stops right there. That is the name of this. Then from here, Adelpha phylaca goes down to here. The difference between these two species of butterflies, this one and this one, is probably only one base pair or maybe two base pairs. That's why there's a case here where there's a mix. There's one individual here and there's another one there. That's probably right at the very tip of the barcode. Now these two things then have got very, very similar barcodes. But look how different the butterflies look. What that tells you is these split up from one common ancestor not very long ago. But there was very intense selection for this color pattern or this color pattern. And in fact, these are both mimics of big complexes. So this is a mimic of one big complex and this is a mimic of another big complex. So the selection was very strong to change the color, but not the barcode. The barcode, in fact, is very involved in your own oxygen metabolism, or the oxygen metabolism of this butterfly. So if it mutates in any serious way, butterfly dies. Here's the caterpillars of those two. See how similar they are in color pattern? And they eat almost exactly the same plants. So these have only been apart probably for a very short time. They've only been separated evolutionarily. They co-occur, they go side by side, and eat the same plants. But the point being is that they have just differentiated a little bit on things like food plants and their barcodes. But because each one of them has been selected to be part of a big mimicry complex, that pushed their change very, very rapidly. Now, how do we find these things? This is a set of barcodes for a bunch of butterflies. Each one of these is one butterfly. And the length of this line tells you how different this set right here is from that one, and that one, and that one, and that one. So you see how these are very short lines here. So that tells you that these four right here are very similar barcodes and almost without doubt are the same species. But right here are two which have the same classical names. In other words, these are names put on by eye. You look at them and you call them Talides sinois. But look at the difference in the barcodes. Very long. This is a big difference. That's probably 3-4% of their barcode is different from these two. That tells you, whoa, this is probably two different species here. Well, here's an example, a concrete example that we followed up on. So here, these things here, this thing right here, Parachartes filetes. There were two specimens by eye called the same species, but look at the length, how separate they are. See, they're not at all like this little cluster that you see right here. Incidentally, when you see something like this, this is an aside, but you see how there's 1, 2, 3, 4, 5 of them here with the same name, and then with a very different name. That tells you there's an accounting error. In other words, this specimen is almost without doubt a Thracides phyton. But the people doing the management of the information put this name in the wrong box. Okay? Because the specimen itself is what gave this result here. Anyway, we have this Parachartes filetes, which has a long barcode. So we go out and catch more of them, see, because this becomes a warning signal. Now, taxonomy now is telling you, hey, get more specimens of that. So you do so. So we've got this, here's Parachartes filetes. Now we got all that many. And immediately you discover that here's a group separated by a moderately long line, which is there. But there's something suspicious. This little group right here has got this like a beginning of a separation from this group down here. So that says, go get more. So of course, remember I showed you the photograph of the parataxonomists in the very beginning. The instruction goes out to them. And they say, okay, they go and they look in the records, they see where these caterpillars were caught, what they were eating. And they read the forest. They go back into the forest, look for those plants, and then look for those particular caterpillars. So we get more of them. And it turns out that immediately they come back at me and say, hey, all this group down here is eating grasses. And this group up here is eating grasses and palm leaves. So I say, go get more. So they go out and now look how many we have. All of these. And look what happens. This entire group, very tight line, eats grasses deep in the shade of the rainforest. This entire group, down to there, eats palm leaves in the deep shade of the rainforest. This group right here eats palm leaves also, but a different genus of palms. Same side by side, but different genus of palms. And this group down here eats grass, but on the edge, broken up rainforest and in dry forest. So it's a sun-loving species. This one up here is a shade-loving species. Well, wow. So now we say, okay, while you were doing the collecting, you also photographed the caterpillars. I'll look at the caterpillars. Turns out these are the two who eat the two deep forest palms side by side. These are the two that eat grasses. And look at the color differences. These look more like a grass blade. Pale green stripes. That's what grasses do. Both of them. But one's from deep shade, the other's from the sun edge. These are both palm eaters, very deep shade. And look at the size of the head. Big head, these are all the same age. Big head, small head, small head. What does that mean? The head of a caterpillar is full of muscle. That's what the head is for. It's the anchor for the jaw muscles. So the mandibles, which chop up the leaves, in this guy will be much stronger than they will be of this guy. Why? Grasses are tender. Palm leaves are very tough. So what you do now is as these have separated, we've selected for bigger heads, more muscle in the head, better mandibles for cutting tougher leaves. And you get different color patterns that go with them. But again, when we go back to this thing here, still, they're all very similar. And if you didn't have big samples like this, you might say, oh, that's just variation. Well, it turned out that in doing this, it turned out that that one right there is the one that had a name for about 200 years. And all three others have been lumped under this one name. So now at this point, my job as if you like, an administrator, an organizer, a coach or cheerleader for this kind of thing, is to say, I now need to go and sweet talk the taxonomist into taking his or her time, in this case it's his time, to stop doing what he wants to be doing in that museum full of all those specimens and describe, put names on, do the taxonomy of the 3 new undescribed species. So now we can then write articles about them, we can talk about what they do, and we can think about how they can be used or not used, or are they endangered, are they not endangered, where do they occur? So of course, fortunately, this is just in one place in Costa Rica. What you discover when you do this is that each one of these has a range, Mexico to Panama, or Mexico to Brazil, Venezuela to Argentina. And so you begin to suddenly open up the book that they are and begin to read it. And because you have names on these things and because you have the barcodes on it, you can put that information on the web. which is where it is now. So you can put that information on the web, and somebody in Brazil, in Venezuela, and Colombia, and Mexico, and South Texas can make use of it themselves as well. And write back to you and say, whoa, we don't have a case like that in near Laredo, Texas. We got something else going on, which is very similar. How do we relate? I say, well, you got to get something from Tamaulipas in Mexico or something from Veracruz in Mexico and fill in the gap between Costa Rica and Laredo, Texas. And so now we can have a conversation about these species. Whereas if you didn't have names and you didn't have the names correctly applied, you couldn't even have that conversation. So we're stopped. We got about 1 minute to go here. So we're going to stop here. Now, oh, these are the adult butterflies, just to give you an idea of those 4. Imagine somebody comes along and says, oh, you can identify things with binoculars. No problem at all. You tell me how to tell those species apart. We have to stop right there. I didn't actually get to the end. We'll pick up right here and continue on Tuesday. Okay. All right. So there we go. We stop for the moment. Thank you.